Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Today we are going to cover part 3 of Learn Bioinformatics from the Scratch. Before this, I have covered about introduction of sequences and then we proceed towards the multiple sequence alignment. In today's session, we will cover somewhat or some introduction to the concept of homology. So let's start. If you're working in bioinformatics, it means you should have some familiarity with sequence alignment, with phylogenetic trees, and all those things, or broadly speaking, in evolutionary biology. To cover these things, you should know the concept of homology or similarity. So, what is homology? Homology is actually it explains the similarity between two sequences, whether DNA sequences or RNA or protein sequences, or I would say amino acid sequences. So, the similarity between these two sequences will be called homology. But if we call two sequences homologs, or if we are accepting that these two sequences have homology, in that case, that means that these two sequences share a common ancestor or share a common ancestry. It means these two sequences came from a single ancestor. But how we will call, like, what is the boundary of homology and all these things, we will discuss in next slide. What are the application of homology in bioinformatics or in evolutionary biology? So first of all, homology identify the evolutionary relationship. For example, if two sequences are homologs, that means these two sequences are sisters, are brothers, whatever you call it. Okay, their parents are same. With this logic or with this hypothesis, if two sequences are homologs, so we can say that both these sequences must have similar function. So by this way, we also allow homology also allows us to predict the gene function, and by this method or by this concept, we can also infer structural informations like homology modeling. We will cover on later on, or some functional similarities. So overall, to conclude this slide, homology just tells us the similarities between nucleotide sequences or amino acid sequences. And if two sequences are homologs, then we say or we accept that yes, these two sequences are coming from the same ancestor. Okay. Is there any practical threshold to decide what is the threshold value of homology? So in practice or in research scenario, sequence with greater or with more than 25 to 30% amino acid identity in proteins, actually we will call it homologs. But on the other hand, in case of DNA and RNA, a threshold greater than 70 to 80% of identity is regarded to homology. Actually, in theory, for protein sequences, this is 25%, but I explained the term 25 to 30, and exactly the 25% region, we call it twilight zone or twilight zone. And above then 25 to 30%, we can safely call it homologs. And here more than 70 to 80% of identity. If two sequences are containing or retaining, then we call it they are homologs. Okay, based on homology, or based on the definition of homology, we have three different types of more kind of classification that develops. The one is called orthologs, second one is called paralog, and the third one is called xenolog. What is actually the ortholog? Remember, ortholog, paralog, and xenolog, all these three are homologs to each other. You can say this way, like these are the different subtypes of homology. So first of all, the sequence must be homologs, the sequences must be homologs to each other. Then we will further decide, is there an ortholog or paralog or xenolog? So what is the difference between orthologs? So let's discuss. Those genes are those sequences which are present in different species. This is a kind of characteristic that these orthologs will be in different species, but they are originated from the common ancestor. But how if they are in different species but they are originated from the common ancestor? So they actually originated in the common ancestor based on the process of speciation, or we also call it divergence. So when the process of speciation takes place or gene divergence takes place, so to that phenomena or to explain that phenomena, we call such sequences orthologs. What is divergence or what is speciations? For example, you are walking on a straight road and suddenly you have two paths now. So you went or you take the right path and your friend take the left path. So actually you both diverge into different paths. But before diverging to different paths, you both were on the same route. But in this case, in my example, I assume two people, you and your friend, but in case of sequences speciation, they are not two, they are kind of one sequence, but with passage of time, it experienced some mutation, some more mutation, some more mutation, and a time will come that a new genes is emerged, and that genes shifted to some another species, and that's why both these genes, they are speciated, they are diverged from a single gene, and now they are in different species and they are homologs. So such kind of genes we will call it orthologs. For example, beta globin gene in human, this is also present in human and it is also present in mice. This is a classical example of ortholog, and it is believed that before, back in the history, it was a single gene and that was present in rodents and primates. But and later on it was diverged or speciated into mice and human. So, what are the characteristics of orthologs? Number one, they typically retain the same function in different species. If in human the globin gene performs some certain functions, it means it will also its ortholog in mice will also perform the same function. And they are used to study evolutionary relationship and infer the function of gene across species. So I hope you understand uh, orthologs. Now, what is paralog? genes or sequences within the same genome that originated from the common ancestor. But in this case, they are originated from a process which is called gene duplication. Previously, in the case of ortholog, the gene was speciated into two, and one gene retain one species and another gene retain another species. But in case of paralog, the gene is divided into two. I would not say divided into two, but it duplicated, just like in PCR we are amplifying genes. You can say that way, because division is something else. So that word I'm taking back. It's not divided actually. It's duplicated. Okay, so sometime a gene is duplicated with the passage of time, and then the same species, the same genome, it contains two copies of their gene, and with the passage of time, the body adjusted, the function adjusted, and then uh, there are, or we see, two similar genes in the same genome. So to this phenomena we call it gene duplication, and genes or sequences arises with this gene duplication, so we call it paralogs. What are the examples? The example are once again alpha globin and beta globin. Let me give you an example. You must know about the hemoglobin of human hemoglobin. So the human hemoglobin is composed of two alpha and two beta globin genes, alpha and alpha dash, some people call it prime, and beta dash or beta prime. So both these both these genes, or both pairs of these genes, it means two alpha and two beta, they combines, they attach to the heme ring, or at the center, and they makes a complete unit, we call it hemoglobin, which carries the oxygenated blood. Okay, so in that case, if you see both the alpha and the alpha dash, they are present in the same genome of human. So in this case we will call it that the alpha and the alpha dash, the beta and the beta dash in the globins, they are actually paralogs. Okay, what will be the characteristics of paralogs? They often evolve new functions and specialize in different aspects of the original function. Yes, of course. If you if you ask me how it evolves, because the primitive version, if I would say myoglobin, they do not it does not contain the four set of genes, just like hemoglobin has. So that's why myoglobin is not so specialized in function as hemoglobin is. And the second one is that they are important for understanding gene families, expansion, and functional diversification, like how genes are expanding, how genes are, with the passage of time, they are making new functions, they adopt new functions, or they are specializing new functions. Um, you can say like that. Okay, so what is the third part? The third part is xenologs, and xenologs are those sequences that are similar, but they are transferred from one species to another by HGT. To the HGT, we normally call it horizontal gene transfer, and it took place in different species, just like, for example, I would give you a basic example. Okay, have you ever heard the name NDM1 or New Delhi metallo beta lactamases? This gene was initially identified in a patient, a Swedish patient in 2009, and then it actually hydrolyzed the beta lactam antibiotics, or you can say it resists the beta lactam antibiotics. What are the beta lactam antibiotics? Those antibiotics which contains the beta lactam ring, okay, like penicillin and others. Most of the beta lactam antibiotics are those which contains the lean classification or the naming criteria. So xenologs actually they takes or they shift it to another species directly by the HGT. Okay, what is HGT? It's just like I was talking about that patient. So the gene which was initially discovered in 2009 or identified in 2009, and later on the same gene was shifted to other species, even it was found or it was observed or identified in the non-pathogenic E. coli. So how it went there from pathogenic, let's say Klebsiella pneumoniae to the E. coli, how it's been there. So it's because of the horizontal gene transfer. Because if you if I give you a scenario, bacteria has another kind of genome. One is present in the nucleoid, the normal genome, but there is also a plasmid, the double-stranded extra chromosomal circular DNA is a plasmid, and the plasmid is self-replicating. So at times, I would explain it this way: at times, if a pathogenic bacteria is dropped on the floor with the non-pathogenic bacteria and they divides and redivides, so there are chances that the plasmid will shift from one pathogenic bacteria into the non-pathogenic bacteria, and that's why the non-pathogenic bacteria will, or the next generation of non-pathogenic bacteria will acquire that property, and that non-pathogenic bacteria will also be able to resist antibiotics. So in that case, both these two genes, one is in, I would say, as per my example, if you identify in E. coli, and in another case, it is also present in Klebsiella pneumoniae, and in another case, if it is also present in Staphylococcus aureus. So in this case, this gene is actually shifted into these species by the process of horizontal gene transfer, and we will call such genes or such sequences xenologs. You cannot call it paralogs or orthologs. So what are the characteristics? As I explained already, they acquired through mechanisms like bacterial conjugation, transduction, endosymbiosis, and they can introduce entire new function into a species. How? As I explained to you earlier, like a non-pathogenic bacteria will become a kind of pathogenic because they got the ability to resist the antibiotics or the beta lactam antibiotics now. So this is a specific case of xenolog and how they come to the pool. I hope you understand the concept of homology and the paralogs, xenologs, and orthologs. So our next session will be what is BLAST and how to use it. So we will carry on this session, and hopefully we will enter into the some advanced stuff, and let's see how long or how much we can cover it. Thank you so much for now. If you are liking these sessions, please tell me in your comments and share it with your colleagues, friends, and students. And thank you so much. Bye bye.